Hello, in this podcast we're going to be learning about how traits are inherited, and our learning objective is that we will understand why sexually reproduced offspring resemble but are not identical to their parents. We're also going to be learning about gel electrophoresis and pedigrees in this podcast.
This land is your land, and this land is my land.
This song was sung and composed by Woody Guthrie. He was a famous folk singer.
From the redwood forest to the Gulf Stream waters, this land was made.
I'm bringing him in here right now because he suffered from a genetic disease that eventually killed him called Huntington's disease. A person who has Huntington's disease, and fortunately it is a rare disease because it is a horrendous disease. A person who has disease is completely normal until they reach the age of 34 to 44. The first symptoms that they have are physical. They have random jerky movements. Eventually the memory becomes affected. As the physical symptoms get worse and worse, they gradually lose control of their body, and also their thinking becomes impaired. They eventually get dementia, which is, in this case, can be somewhat similar to senility or Alzheimer's. And also they get psychiatric disorders like anxiety and compulsive behavior. And people who have this disease frequently are put into institutions. And like Woody Guthrie was institutionalized, and he spent the last year of his life and eventually died in Creedmoor, which is not too far from here. It's a fatal disease because they lose such control of their body that they're really unable to even swallow. This, if you have the gene for Huntington's disease, you will get it. Now, the way they test this is by a process called electrophoresis. Now, the first thing with gel electrophoresis is that the gel is made of something called agarose, which comes from seaweeds. In this electrophoresis we do something that's sort of similar to that in our lab, where we do paper chromatography. Remember we did a lab where you saw the pigments in a marker become separated. The structure of DNA is what makes gel electrophoresis sort of possible, because the backbone of the DNA contains phosphates, and phosphates tend to be negatively charged when the solution is neutral. So if you put an electric current across the gel, that will make the DNA migrate towards the positive charge. So this is basically what the gel electrophoresis sort of looks like. And over here is where you put the segments of DNA, and first you chop up the DNA into small pieces, and you put the pieces in the wells, which you see here. And we're going to be modeling this in a lab in a few weeks. And you basically put a charge in there, and they get drawn towards the positive part. And then what happens is that the gel that it's in, the agarose gel, acts sort of like a hurdle that these things need to cross. Like the green things here are the molecules of the agarose gel, and the blue are different sized segments of DNA that you may see in the Huntington's gene or in any other type of gene. And when you switch the current on, this is what happens. See, the smaller segments, they're able to travel much faster than the longer segments because the longer segments get caught more on the agarose. This is what happens when you run the gel. You start off with the DNA all in one place, and you see you've got different sizes, and they're all put together. Then when you turn the current on, the smaller ones go faster than the larger ones. And then you end up with bands going across the gel, and that's what you use to identify. You won't see the actual molecules, but you can see the bands. And we will be doing a number of labs where we're going to be simulating gel electrophoresis, and we'll be identifying things with the bands, and that will include the State Lab of Biodiversity. But it's quite possible for you to see a gel electrophoresis diagram on the Regents exam. And I have seen them in the past, but they're not in every one. Now here's some vocabulary that you need to know for the remainder of the podcast. Some of these you might have seen before. A gene is a section of DNA that codes for a protein. And remember, we saw that in an earlier podcast, going from how a gene goes from DNA to RNA to protein. An allele is a version of a gene. Some genes have several different versions in them. For example, with eye color, there's blue eyes and brown eyes. Those are the same gene, but different versions of the gene. Genotype is what the individual's alleles are, or what the allele is. That's because the reason I put down plural and singular in there is because we have two chromosomes, so we have 2 copies of every gene. Those copies could either be the same copy or they could be different copies. And if they're the same copy, then that's called, if they're the same allele, that's called homozygous. If they're different alleles, that's called heterozygous. And now you don't actually see the genes, instead you see the trait that comes out. And that's called the phenotype. And the phenotype can be modified and frequently is modified by the environment. For example, sticking with eye color, you see my eye color can change from day to day, and that's caused by environmental things. Also, if you look at identical twins, like here are 2 identical twins who are former students of mine, you can see that they don't look exactly the same. They have different birthmarks on them, and that's all caused by the environment. It's not caused by their genes at all because they have identical genes. So they have slightly different phenotypes. The alleles can be labeled as dominant or recessive. So that if we stick with the eye color example, the gene is a gene that codes for melanin in the eye, and melanin is what gives it the brown color. And if you have the allele that works, where the gene gives you the protein for melanin, then that's going to be a dominant gene. You'll need 1 copy of that. 1 working copy will give you the protein melanin, and you'll have brown eyes. So brown eyes is known as a dominant allele. On the other hand, if you have the allele which has a broken version of the gene for melanin, in other words, the protein that's coded is one that doesn't work. At some point there was a mutation, and that mutation basically just made that gene defective, so it doesn't make anything that works, so that melanin isn't made. Instead, if a protein's made at all, it basically doesn't do anything. And that gene is known as recessive. If you have the heterozygous version, where you've got 1 copy of working gene, 1 copy of a non-working gene, it works. You got the melanin, you got the brown eyes. It's only when you don't have a working copy that you don't get the melanin, and then the person has blue eyes, like I do. Also, a pedigree is a chart that shows how an allele is inherited in the family. I know you don't know what it is just from here. We'll get to it later, where you actually see an example of a pedigree. Now here we're going to look at one of the first genes that people looked at in terms of inheritance, in terms of dominant and recessive. And this was done by Gregor Mendel back in the 19th century, mid-19th century. And he was looking at pea plants. Here the gene is for pea plant height. 2 alleles, tall and short. So that he took a plant that was completely pure for tall, another plant that was completely pure for short. In other words, both of them, the tall one came from a line where you only had tall plants. The short one came from a line with only only short plants. So both of them are homozygous. Mated them together, and normally you would expect them to be somewhere in between. But what happened is he ended up with all tall plants. So how did he end up with that? Well, we'll see in just a little bit. And then he took those 2 second generation ones, mated them together. And what were the offspring like? The grandchildren? Well, 75% of them, 3 out of 4, were tall. And 25%, 1 out of 4, were short. How did this happen? Okay, here we have our first generation, tall and short. Now, as I said, we're dealing with 1 gene with 2 alleles. Now, keep in mind that you may think, oh, this is like this with people being tall and short. It isn't. This is 1 gene. In people with height, I think we're dealing with 170 genes. So it's much more complicated than this. So here we're starting off with big Y, big Y. Now, capitals denote the dominant, lowercase denote the recessive. So right here we have our homozygous two big Y's. In the gametes in meiosis, they can become separated. So you're going to end up with one gamete with just a Y, big Y, another gamete with little y here, big Y here, and from the short plant you've got two little y's. Let's look at all the different combinations that we can get here. We could have the little y over here. So let's draw our little y. Match up with the big Y here. And that gives us our big Y, little y. This one's heterozygous, so that gives us a tall plant. So 25% of these will be heterozygous tall. Now this big Y doesn't have to meet up with the little y on the left. It could also meet up with the little y on the right. This one here. When it meets up with that little y on the right, then we end up with the same thing, big Y, little y, which is heterozygous, and we just go with the dominant one, so it's a tall plant. And this little y over here could meet up with the other big Y. And that gives us the same thing again. But this big Y here could also meet up with the other little y. And we end up with yet another heterozygous. And these are all tall, 100% tall. And that's precisely what we ended up with in reality in Mendel's experiment. There's a quicker way to actually do this, and that is something called a Punnett square. And for this, we're going to look at the other generation, where we put the parents on the axes. For example, we will put one parent here. Remember, we'll do one parent in red, one parent in blue like before. So here's our big Y, little y for one parent. And now we're going to do big Y, little y for the other parent. So a quicker way to do it, because like here, now we have them divided up into the gametes. Here's one gamete, another gamete, one gamete, another gamete. So one sperm, so they're either sperm or egg, doesn't make a difference which is which here. And then basically we take this one here and we just put them down in each square here. So here's our big Y and our big Y. I'm just putting it over here because just with convention we tend to put the big Y's before the little y. Here's our, and do the same thing here, little y down here, little y down there. So let's just go straight down where this goes over here, and this goes down there. And then you take the ones on the side, and you just do the same thing. This goes across, and this one goes across. So this little y goes here, and also over here. I'll put this here, so this big Y goes in here, and across over to here. Okay, so here are the four possibilities. We have for our third generation, we have one box that has big Y, and that's tall. We have two boxes that are big Y, little y, which is still tall. And then we got one box, which is little y, which is short. And since there are 4 possibilities, each one has a 25% chance of occurring. So we'll end up with 75% tall, 25% short, which is what Mendel saw happen to the peas. And that's how you figure out how one particular gene gets inherited. Things get a bit more complicated when it comes to the assortment when you're looking at genes together, because it's the chromosomes that become inherited. Chromosomes are basically sorted by meiosis, not the genes. So, in other words, genes that are on the same chromosome stay together. In other words, they are linked. That's what they actually call it, linked. They can still be shuffled. These genes, though, shuffled by a process known as crossing over, also known as recombination. Remember, we learned about this before when we were learning about meiosis back in the reproduction unit. And the further the genes are apart, on a chromosome, the more likely that they will be shuffled. Now here's a pedigree. This is a diagnostic tool that's frequently used to see how a trait will be inherited and to see if you have the chance for a trait. So I suggest copying this stuff into your notes, and it tells you the nomenclature of it. And this particular gene that's being shown is a recessive gene. The way you can tell is you see the half-shaded circle there means the person is a carrier. If they're a carrier, that means that they have the gene, but they don't show the trait. For example, if we go back to those pea plants, the pea plants that were heterozygous, where they had both the tall gene and the short gene, while they were completely tall, you couldn't tell, just looking at the phenotype, there was no difference between a heterozygous tall and a homozygous tall. But the heterozygous tall was a carrier for short. So if you look at these parents right here, even though the mother here is a carrier, that mother doesn't show it at all. And you can tell she's a carrier because she has a son who shows it. Though I'll tell you that you sort of can tell from that if that person has it, that means that both parents would have to be a carrier. So there's actually, looking at this over here, the only way you could have something like this happening would be if the gene is on the X chromosome, because the male only has one X, the female has two X's. So that since the male has only one chromosome, even though it's a recessive gene, if it's on the X chromosome, then the male would show it. And here's a place where you see that. This I put down as a famous pedigree because the people in it are very, very famous. And what happened is, you see this person here, King Edward. He had a mutation. And for a mutation to be heritable, it has to occur in a gamete, which it did in Edward. It occurred in a sperm, and not just any sperm, but the sperm that fertilized his wife's egg and became Queen Victoria, who became a carrier. She married Prince Albert, and they had lots of kids. And they had one son who first showed the trait. None of their daughters had the trait, and actually none of their descendants who were daughters had the trait. This trait is hemophilia. Hemophilia is basically, it's a genetic disease where that person is a bleeder. Their blood does not clot. So that until we found a good treatment where we give the person a clotting factor so that their blood would clot, these people had to be very, very careful, and frequently they died in childhood by bleeding to death because a minor cut, a minor bruise, it just keeps bleeding and bleeding, and they bleed to death. The aristocracy of England, in order to form alliances, married people all over. Then they ended up spreading this disease throughout Europe's aristocracy. World War I was a battle, in a sense, between Victoria's grandchildren, and it was all between it was in a sense you could look as a family squabble. England had King George. Nicholas was the Tsar of Russia, was Victoria's grandson, and they had a child, son Alexis, who has hemophilia. Though hemophilia actually was not did not cause his death. He was, just because you have a disease that can cause your death doesn't mean it will. What happened to Alexis was, when the communists took over, they killed the entire royal family, so he was murdered. On the other side of the war, Kaiser Wilhelm was also Victoria's grandson. And if you go down to the bottom, you'll find that Victoria's English branch, where we are basically free of hemophilia. And down here, we see Elizabeth II, that's the current Queen of England. And number 11 is the Prince of Wales, Prince Charles. Number 12 is Diana, and below there we have their two children, William and Harry. Now we come to our concluding questions. Number 1, how many copies of each gene do you have? Number 2, what is electrophoresis? For number 3, let's look at this picture here. And this picture shows my family when I was 23 years old. And here I am. Look at my eye color. You see I've got blue eyes. Now here's my mother, and she has brown eyes. My father also has brown eyes. And my brother also has brown eyes. Which allele is recessive? So which allele is recessive, brown eyes or blue eyes? How can you tell? Number 4, here we have a chart. What is this chart called? And for extra credit, I want you to complete this figure to show the inheritance of the blue eye allele in my family and identify the 4 of us. And I'll see you in class tomorrow.